Presented by Caltech.
So Stuart received his medical degree, the Doctor of Medicine, from the Hahnemann Medical University Hospital in Philadelphia in 1973. And he then moved on to the Department of Anesthesiology at the University of Arizona, where he has been a professor and has practiced clinical anesthesiology for about 45 years now. While at Hahnemann, Stuart got interested in the role of microtubules, the role that they played in cell division, and that led him later on to speculate that microtubules may also play some role in consciousness. As some of you might know, in 1989, Sir Roger Penrose wrote his book on consciousness called The Emperor's New Mind. And that's where Penrose argued that consciousness needs to be fundamentally non-algorithmic, and therefore quantum processes need to be involved or invoked in order to explain consciousness. So Stuart got inspired from his book and wrote a letter to Penrose saying that perhaps microtubules are those sites of quantum processes. And later on, Penrose and Stuart, of course, came up with this theory of orchestrated objective reduction, or Orch-OR, which could link between the brain and the space-time geometry, something that is fundamental to the universe. The Orch-OR has been widely criticized, I must say, and as a result, the theory itself has evolved over the past 25 years. Nevertheless, Stuart keeps reminding me and cheerfully saying that it is better to be criticized than to be ignored. So again, today now, Stuart will be presenting on or telling us how anesthetics can link between quantum vibrations and microtubules. And he'll also tell us why Orch-OR should be taken seriously. So let's welcome Stuart Hameroff.
Thank you, Kunal. Thank you all for being here. It's a great honor and privilege. What you see on the slide in the background is a neuron, electron micrograph, immunofluorescence. And you can see the surface membrane. I don't know if this pointer's working, but you can see the edges, which are the membrane. You can see the nucleus, which everybody focuses on, the membrane in particular. And in the back, inside, you see in the red actin and the yellow microtubules, or tubulin. So main point is to go inside the neuron to find out neural processing, including consciousness. Now, consciousness is a controversial subject. We take it for granted that when we open our eyes, the world out there appears inside our heads. Bing, as I like to say, this implies experience, qualia, having subjectivity, first-person point of view, phenomenal experience, conscious awareness. And that's the trick. And how do we explain it? Well, there are a number of approaches, as you can see, starting upper left. Neuroscientists tend to probe the brain with electrodes and so forth. AI and roboticists, artificial intelligence people, try to simulate brain processes in machines. Artists capture the essence of consciousness. Physicists ponder the nature of reality, Schrodinger's cat. Psychiatrists, the inner subconscious. Anesthesiologists that look suspiciously like me ponder how anesthesia works. Meditators and Western philosophers all approach this problem, this issue of conscious awareness. Now, the point I want to make is that anesthetic gases selectively erase consciousness-sparing, non-conscious brain activities. We do a lot of anesthesia for spine surgery, for example, where we want to make sure the surgeon isn't damaging the spinal cord. So we monitor electrophysiological evoked potentials, visual, motor, sensory evoked potentials in the brain. So signals are getting through, but there's no conscious awareness. The patients are unconscious or anesthetized, but their brains are quite active. Therefore, the precise mechanism of anesthetic action may illuminate the origin of consciousness in the brain. So what distinguishes conscious processes from consciousness is what goes away with anesthesia. Now, in the year 2000, this book appeared on the greatest inventions of the past 2,000 years. And as you can see, anesthesia is at the top of the list, at least alphabetically. And I wrote the article, and basically what I said was, imagine going through life today in our modern world without anesthesia. You know, you broke your leg or your appendix burst or something like that. You'd face surgery without, you know, in a lot of pain. But anesthesia allows you to basically go on vacation while your body's in the shop, more or less. So historically, gas anesthesia started in the 1800s, 19th century. For example, laughing gas, nitrous oxide, and at low concentrations, it's euphoric, giddiness, and I don't know if you can read the caption. It says prescription for scolding wives, which probably isn't politically correct these days. And ether frolics, and small amounts of breathing ether made people giddy and happy, and parties were devoted to that. Now, somebody realized that if you go deeper with more anesthesia, you became unconscious and unresponsive. And H.G. Morton developed this as a tool for surgery and would render people unconscious. And this is a famous demonstration at Massachusetts General Hospital in 1846. Now, I've been doing this a long time, but I'm not in this picture. I accuse some of my friends of being there. Now, moving ahead, this is a famous, at least in my neck of the woods, procedure of the first bridge to transplant in the 1980s, in the early days of heart transplantation. And the surgeon in the middle there was Jack Copeland, who was our heart surgeon, and he had been doing a lot of transplants and developing an artificial heart. And the Jarvik 7, none of those were available. And this guy, a 30-year-old guy, had fatal cardiomyopathy. He was dying, and Jack and his colleagues had this device. You can see it in the first guy's hands that they had built in the lab and been using on pigs, but not in humans. Definitely not FDA approved. Jack said, well, the guy either dies or he gets this device. So they put it in, and he survived a month, at which time a real heart came in that would match. And in the middle of one night that we took him to the OR, took out this device, and there's Dr. Vance actually pondering it and looking at an empty chest and figuring out, will this heart actually go in there? And it did. And the guy lived another 9 years pretty productively. And there I am in the background in my younger days peering on. So anesthesia, anesthetic gases, it was discovered in the 19th century that breathing certain gases mixed in air or oxygen, you need air or oxygen, of course, immobilizes or anesthetizes all animals studied. Mammals, amphibians, insects, fish, worms, protozoa, even plants are sensitive to anesthesia in terms of their tropisms, moving towards the sun and that sort of thing. Now, interestingly, the gases varied greatly in chemical structure. They included ethers, halogenated hydrocarbons, nitrous oxide, and even the inert element xenon. Now, the inhaled concentration at equilibrium required to anesthetize or immobilize animals differed among gases, but was the same for each gas for all animals. At equilibrium, the same amount of diethyl ether, for example, would anesthetize a fly and a horse. Once at equilibrium, the same concentration. They differed among the anesthetics, but for each anesthetic it was the same for all animals. This became known as the anesthetic's minimum alveolar concentration, or MAC, inversely related to potency. The more you needed, the higher the MAC, and the weaker the anesthetic. Now, a common factor was desired to try to put this all together related to potency, and this was sought for the disparate chemical structures. And scientists tested solubility of anesthetic gases in various solvents and compared the results to MAC. Now, I should say that these compounds were chemically different. There you see xenon, a single atom, and we saw the ball and stick figures, but they had something in common in that they all had at least regions of a completed electron outer shell. That is a full complement of electrons, therefore no polar charges, no chemical reactions occurred, and they were basically chemically non-reactive, just with this completed outer shell, and xenon is a complete outer shell. Now, when we have such a situation, molecules and atoms with full electron outer shells don't react chemically, but attract each other and then oscillate by quantum van der Waals London forces. Electrons induce dipoles in nearby electron clouds, which induce dipoles in original electron clouds, and the dipoles then attract. And they're happiest at what's called the van der Waals radius, where basically the electron clouds just basically touch each other. And you can see at the bottom that one induces a dipole on the other, and the other induces a dipole back. So, and then they oscillate, but I'll come back to that in a second. Now, scientists tried to look for where these anesthetics were acting in the brain in terms of their solubility. So oil and water don't mix, and these are nonpolar and oil-like. So if you think of a human body ground up, maybe think of somebody you don't like, ground up and put in different solvent compartments, you see here that going from lower right, northwest sort of, increasing polarity. The other axes are permittivity and dielectric and stuff like that. And you can see certain compartments that correlate with solvents shown on the right side. So two scientists working separately in England and Germany, Meyer and Overton, between the two of them covered pretty much every gas known to cause immobility or anesthesia in a variety of different animals. And they looked at different solvents and found this amazing correlation over many orders of magnitude between the potency, that is the MAC on the right, as you go up, they're weaker because the MAC is higher, versus solubility in olive oil. Olive oil was the solvent that best mimicked the potency, the solubility in olive oil matched the potency of an anesthetic. So methoxyfluorine is the most potent anesthetic, about 0.25% will anesthetize people. And nitrogen is a very weak anesthetic, requiring over 10 or 20 atmospheres. So at that high concentration, even if you have oxygen, you lose consciousness. And nitrous oxide is at about 1 atmosphere. So it can anesthetize you, but not completely, because you still need oxygen. So this amazing correlation over many orders of magnitude suggested that the anesthetics were acting in the little circle of aromatic rings, which best correlates with olive oil in the lower right in a highly nonpolar environment. Now, anesthetics are poorly soluble in water and blood, and when you breathe them, they go into the lungs and into the blood and quickly escape the lungs for more olive oil-like material, which includes fat and nonpolar regions in the brain and throughout the body. And you can see that that region correlates with solvents, dimethylbenzene, methylbenzene, and benzene, so which are basically the same as aromatic rings in certain amino acids, tryptophan, phenylalanine, and tyrosine. So these aromatic rings, or benzene, came out of chemistry in, I think, the 1700s. Chemists knew about the alkanes and the alkenes, and they had a compound named a C6H6 with different properties, and they didn't know the structure of it. The alkanes and alkenes are basically linear molecules, and Kekule, a German chemist, had a dream one night that one of these chains were snakes that swallowed its tail, and he woke up and said, haha, C6H6 is a ring, and he turned out to be correct, and we now know that that ring, shown on the bottom right, has 3 extra electrons, unpaired electrons, which form electron clouds. So, and anesthetics bind to these types of compounds by van der Waals London forces, specifically in a region of the Hildebrand solubility coefficient that you see there, indicating a nonpolar environment with these pi electron resonance clouds, as in these ring structures. So pi electron resonance clouds seem to be where anesthetic action was occurring. Now, we also know that these pi resonance clouds, for example, 2 benzenes by themselves, and I should say that olive oil, if you put it all together, it's a mess, or like benzene is basically gasoline. But if you put these aromatic rings at specifically geometric arrays, like in graphene, for example, you get interesting quantum effects. So that's more of what I'm talking about rather than the bulk effect. So we know that these attractions occur, and once they occur at the van der Waals radius, the dipoles couple and then oscillate at about 10 to the 12th terahertz. And they can also form quantum superposition, as you see in the right. Now, however, the energy of these paired dipoles is only about 0.25 electron volts, about kT, thermal noise. So this type of coupled dipoles with just 2 benzene rings wouldn't do much. It'd get washed out in the thermal noise. However, collective dipoles with more than 2, and exactly how many more than 2 we're not sure. I'll get back to this later. If you have a collective van der Waals forces collectively in a group of these, the energy is 2.5 electron volts, or 10 times kT. So these become stable, quite stable in a warm environment. So many biomolecules are amphipathic, which means they have pi resonance rings, these nonpolar rings on one end, and polar water-soluble tails on the other. The pi resonance groups attract each other, coalesce, forming what are called micelles, and this was thought by Operin to be the first form of life. Proteins fold in this way with a pi resonance core. These pi resonance groups support quantum mechanisms, including non-locality, coherence, and collapse, that I'll get to later, all implicated in one way or another in consciousness. They're also found in lipid membranes, in the interior of the lipid membranes with cholesterol and other lipid molecules, and in nucleic acids and pi stacks. So all forms of biomolecules, proteins, lipids, nucleic acids, have what we call a quantum underground. That is a core of pi resonance where quantum effects can occur. And this term was coined by my colleagues Travis Craddock and Jack Tusinski in a paper we wrote a couple years ago. I should also add that psychoactive molecules, including neurotransmitters, dopamine and serotonin, and the psychedelics, LSD, DMT, and psilocybin, have extended conjugated pi resonance groups, and they definitely have effects on consciousness. Now here's the hypothesis. Somewhere in the brain, collective pi resonance quantum oscillations critical to consciousness are dampened and dispersed by anesthetic van der Waals couplings. The anesthetic form their own van der Waals couplings, but the idea is that they somehow disperse or mess up collective oscillations shown on the left that would be necessary for consciousness. So I show three benzene rings or phenyl rings. We don't know whether 3 would do it. I'll get later to the point where 86 do seem to do it. So somewhere between 2 and 86, the pi resonance couplings become collective, and you get this quantum state with an energy resistant to thermal noise. And the other prediction would be that psychedelic molecules enhance pi resonance oscillations. So the anesthetics would dampen, and we predict that the psychedelics would increase the frequency. Come back to that point later. Okay, so in pi resonance regions of which biomolecules and cell structures do anesthetics act? Where in the brain do they act? Because they're fat soluble, they go to fat stores, and I often tell my residents there's more anesthetic in the guy's rear end than in his brain, but obviously the anesthetic is acting in his brain, but where in the brain? It's in lipid membranes. Do they act there? Not necessarily. The first study on anesthetic mechanism was in 1846. Claude Bernard was studying amoeboid movement. So these amoeba shuffling along, moving by amoeboid motion. And if he put them in an environment of chloroform, which is an anesthetic, they just stopped. They stopped moving. And when he blew off the chloroform, they started moving again, undeterred and apparently the same. And later, Seifert and Christian showed this effect was directly on the cytoplasm, not via membrane signaling. In the 20th century, we knew that in the early 20th century, I wasn't around then, but that neural membranes are lipids, and these lipid-soluble anesthetics were thought to get into the lipid phase and somehow gum up the works by causing expansion. This turned out to be not the case. A slight variation in temperature would do the same thing without causing anesthesia. Then this guy, John Nunn, whom I had the privilege of meeting, did a study and published in Lancet where he studied effects of halothane, the anesthetic halothane, on actinosphaerium, a small organism with these multiple axonemes. And you can see below that the axonemes were actually double helical or double spirals of microtubules. All those little circles are microtubule on end. And I'll get to microtubules in a second. And they suggested that halothane acted by depolymerizing the microtubules in our brains. Fortunately, that turned out to be wrong because we don't want to depolymerize our microtubules. The depolymerization occurs at about 5 times Mach, at about 5 times what's required to cause anesthesia. But it does show that anesthetics do bind and have some connection to microtubules. Jim Trudell, who is at Stanford, still at Stanford actually, tried to, since it became known that ion channels were important for neuronal firing and depolarization and so forth, he said that, well, the anesthetics get in the lipid phase and extrinsically compress the protein so the ion channel can't open. That turned out to be wrong because, again, a temperature change did the same thing. Finally, in the 1980s, Nick Franks and Bill Lieb in the UK showed that anesthetics act directly in proteins in nonpolar hydrophobic water-excluding pockets, for example, in the membrane-free enzyme luciferase. And they were able to measure emission of photons, and then by adding anesthetics, they did MAC equivalents and found that the more potent an anesthetic was in putting you or I to sleep, the more potent it was in preventing this luciferase. So the anesthetics could act directly on proteins. That was a big breakthrough. So that led to a choice, a fork in the road. Going back to Claude Bernard and John Nunn, do anesthetics act on cytoskeletal proteins, or do anesthetics act on membrane proteins, on excitability of membranes? And almost everybody took the left fork and began to study anesthetic effects on membrane receptors and channels, and they found that the volatile anesthetics do affect receptors for serotonin, glycine, acetylcholine, and GABA. Propofol and etomidate, which are intravenous agent, affected GABA receptors, and ketamine affected NMDA receptors. However, further study showed a mixed bag of conflicting results. And in this editorial in Anesthesiology, the premier journal in our field, Evers and Steinbach concluded that volatile anesthetics were double-edged swords. They both enhance and inhibit ligand-gated ion channels. In other words, there was no common story. A bunch of other papers came out in the early 2000s that showed that various receptors and so forth, it didn't work. None of these showed any consistent effect. And finally, Ted Eager, who was the kind of the scion of anesthetic mechanism research, in this paper concluded that 2 decades of investigation have failed to identify a ligand of voltage-gated channel sufficient to mediate immobility, which is their word for loss of consciousness, since they didn't want to talk about consciousness. Indeed, most targets provide minimal or no mediation. Opioid, serotonin, GABA, glutamate appear to be irrelevant, and no combination seems sufficient. This is kind of a nihilistic paper viewed by many. What do we do now? A new paradigm. Well, what do we do now is go to the right fork, cytoskeletal proteins. And beginning in 2008, Eckenhoff's group at University of Pennsylvania did a comprehensive study finding, for example, that anesthetics bound to 70 different proteins in neurons, about half in the membrane and half in the cytoskeleton. And they then did sophisticated proteomics and genomics and found that basically tubulin was the best bet. They found that, for example, only 3 proteins had genetic alteration due to 2 different anesthetics. The heat shock and the other one really had nothing to do with signaling. So tubulin seemed to be the best bet. They then did a very interesting study with a fluorescent anesthetic, anthracene derivative shown there, again with these pi resonance groups, that is only anesthetic when fluoresced, when illuminated with ultraviolet light. And they gave them to tadpoles, and tadpoles are swimming around, and tadpoles have conveniently transparent heads, so they then illuminated their brain with ultraviolet light, causing the fluorescent anesthetic, and the tadpoles just basically turned over, went belly up. They were anesthetized. They then donated their brains to science, and they found that the anthracene was bound to tubulin, to microtubules, and they graciously acknowledged that this was consistent with our theory that we had published about anesthetic effects having to do with quantum mobility in 2002. They did one more study that's interesting, a clinical study. Taxol, or taxane, is an anti-cancer drug which binds to microtubules and blocks mitosis. And it's used for breast cancer, and they found that the patients, usually women, who came to the operating room for surgery of various kinds, required more anesthetic than other patients. So Taxol, or paclitaxel, increases MAC, and they found there's the binding site on tubulin. The paclitaxel is the one in the minute. A lot of the anti-cancer drugs bind to tubulin to prevent mitosis. So in this paper in PLOS One, we, Travis Craddock and other colleagues, first of all identified all 86 aromatic rings in tubulin, and you can see the tryptophan, phenylalanine, tyrosine, found the anesthetic binding site, and the red spheres are where anesthesia binds. And you can see the paclitaxel in the beta tubulin sits pretty much over that cluster of spheres in the kind of in the middle, slightly to the upper right, near the Taxol binding site. So it could be that the Taxol prevents the anesthetics from getting into their binding site, which is influencing MAC. So taking this all together, anesthetics appear to act in nonpolar pi resonance regions in tubulin, components of microtubules. What do they do there to prevent consciousness? So we embarked on the study that was published in Scientific Reports last summer that I'm going to tell you about now. And in addition to the 8 anesthetics I showed earlier, we added 3 other gases. And these are oddball gases, which were discovered in the 90s, which bind in these same nonpolar regions according to Meyer-Overton, but do not cause anesthesia. These are the non-anesthetics F6 and TFMB, shown in pink, and fluethil, which causes seizures at low concentration, then is an anesthetic. So the question is, what distinguishes anesthetics from non-anesthetics? So we use these 11 gases, 8 anesthetics, 2 non-anesthetics, and then the anesthetic convulsant. The first thing we did was basically do a Meyer-Overton curve and showed that TFMB and F6 have solubility that would put them otherwise pretty good anesthetics, near halothane and sevoflurane, but their MAC, they had no MAC, or the MAC was infinite. We just guessed at 1,000, which would be 10 atmospheres, but basically they don't cause anesthesia. So that's consistent with their known non-action of anesthetics. We then plotted polarizability, that is mobility of the electron cloud for each anesthetic, and the non-anesthetics against solubility. And interestingly, the TFMB and F6 had much higher polarizability than they should, or than the anesthetics. You can see that the oil gas partition would be on the x-axis, but they were too polarizable. And that is, polarizability is the ability to form instantaneous dipoles. So they're kind of too easy. They go along for the ride too easily and might not put drag on the system to slow down the system they were binding to. But polarizability does distinguish anesthetic from non-anesthetic. And here, the same thing for plotting polarizability versus MAC. You can see the two are off the curve. So we then model collective dipole oscillations among the 86 pi resonance groups in tubulin, with and without the 11 gases. Now I say we, this was done by on a supercomputer by my colleagues. I don't do supercomputers, and I'm just reporting the results. I did come up with the idea to do this study and write the paper, but they did the simulations. And so you can see the 86 aromatic amino acids. So imagine they model all 86 and all the interactions among all 86. And what we found was that at KT, at room temperature, a spectrum of collective dipole oscillations with a common mode peak at 613 terahertz. Now each gas added a new oscillation frequency, and that's in the colored ones. So the colored lines are for each anesthetic. I should add that meth is not methamphetamine, it's methoxyflurane, a very potent anesthetic, and death is diethyl ether, unfortunately acronym there. But so each anesthetic had their own, but if you look at the gray lines in the background, that's the native spectrum of tubulin without any anesthetics, and you can see just to the right of the 600 line a density that was this common mode peak at 613 terahertz. Now this is actually 489 nanometers wavelength, which is in the blue-green spectrum. And so these oscillations are occurring in the visible light region. Now, they're not necessarily emitting photons because they stay in, and they're not necessarily excited, they stay internal to the system, but that is the frequency. And these are also consistent with experimental findings in microtubules and tubulin from Anirban Bandyapati's group. Those are the references. Sahu was the first author. And to summarize that work, a lot of work in one slide, they looked at neurons. So in A, we have neurons with nano probes. The solid probes are the pros. Then you can see the neurons. He has 2 neurons. and then in B, a single microtubule with between 2 electrodes, and then 4 pairs of electrodes, and then finally just tubulins with 4 electrodes. And they basically, all of these are insulators, non-conductive generally, but they swept the AC frequency, and they found that at certain frequencies of alternating current input, either the tubulin or the microtubule or the neuronal bundles, or the bundles within the neurons, became highly conductive, possibly even superconductive, ignoring the contact. And they then plotted the resonance peaks where these conductances occurred at each of these levels, and each one showed a triplet, and Honorbahn says a triplet of triplet, kind of like 3 peaks with 3 peaks within one. And you can see they kind of self-repeat at terahertz, gigahertz, megahertz, and then moving up in scale, gigahertz, megahertz, kilohertz, and then megahertz, kilohertz, and hertz, which gets into EEG frequencies. So spanning some 12 orders of magnitude, you get these conductance resonances that all have some kind of self-similar patterns. I'll come back to that in a little bit.
Excuse me?
Yes?
What led you to say possibly even superconductive?
Why did I say that? Because it was highly conductive, and there's resistance between the electrode and the surface. But if you ignore that and just get to where we think the conductance is occurring in the tubulins, in the pi resonance, that could be a superconductor. I don't have any evidence for that, but
Yeah, I was curious about what led you to say possibly even superconductive. Is there some reason for you to think that that region would actually be superconductive?
yes.
What is that?
Because you have collections of pi resonance that are quantum systems. And for example, graphene. Okay, graphene is a quantum material. It's basically these pi resonance put in a ribbon or a chain, and that's a quantum system, and that has superconductivity, and the same thing could be happening inside microtubules.
Well, since superconductivity is a very particular quantum phenomenon not shared by all quantum systems, you're saying that this quantum system shares many key features with actually superconductive quantum systems rather than just arbitrary quantum.
Yeah, well, I should be careful because I'm not a physicist, but and also this is not conductivity from electrons going all the way through. It's actually dipole oscillations. So whatever the analogy or metaphor would be, it's a quantum dipole oscillation that doesn't seem to have that seems to be very efficient and very, very little resistance to it. Okay, the subtraction spectra for tubulin collective oscillations in the presence of gases of each gas show that anesthetics abolished and non-anesthetics did not abolish the 613 terahertz peak. So here's this is the subtraction spectra. So if you start from the spectra for each situation of the tubulin, of the 86 collective oscillations, 86 tubulins oscillating collectively in the presence of the different gases, and then show the difference between the two, the subtraction spectra, this is what you get. And the top 8, the top 2 panels, starting with desflurane, you can see that the yellow, the 613 peak, is missing. It's gone. And similarly, diethyl ether, it's gone. Enflurane, it's gone. Halothane, it's gone. Isoflurane, methoxyflurane, nitrous oxide, and sevoflurane all get rid of the 613 terahertz peak and actually shift energy slower. Now the F6 did not. The F6 got rid of a slower peak, and TFMB, trifluoromethylbenzene, actually increased the peak at 613. And fluethil basically did abolish the 613 peak, but it is an anesthetic. It causes seizures at low concentration, but then is an anesthetic. So the F6 and TFMB, which are not anesthetic, did not abolish the 613 terahertz peak. And the presence of each gas shifted the dominant 613 peak proportional to the potency or the non-potency of the anesthetic. So here we see a plot of the frequency shift at 613 terahertz in terms of how much slower it went. So methoxyflurane, was the most potent and has the lowest Mach, had the greatest decrease, the greatest shift, and all of them fell on this line, and F6 and TFMB were off the line at a high Mach. Nitrous oxide is about 100%. You need about 100% of breathing for complete anesthesia. Of course, we don't do that because you need nitrous oxide. So it was the weakest, and all of these fell on the line. So this is kind of a Meyer-Overton correlation for effects on shifting the 613 terahertz peak. So consciousness correlates with this 613 collective oscillation among tubulant pi resonance groups in microtubules abolished under anesthesia. And as I said, these have been seen in experimentally in tubulin, as I showed you before, the terahertz being the fastest in the lower right. So how would this account for conscious perception and control at brain level? You could say, okay, this is happening at the microtubules. Who cares? They don't have anything to do with consciousness. I would differ. I think what's happening is something like the following, that there's a multi-scale hierarchical resonance system originating in quantum terahertz oscillations in tubulin microtubules inside neurons. So at the far right, you see at the top the dipole oscillations occurring in the conscious state, and below that we've added some anesthetic, and they disperse the dipole and stop the oscillations. So putting the anesthesia aside, just with the dipole oscillations, they can scale up at the frequencies that Anirban discovered for tubulin pathways in rows of tubulins, then in microtubules, up to the pyramidal neuron and bundles of microtubules getting slower and slower, and then cortex and the whole brain, and getting slower and slower. And basically, Roger Penrose, whom I haven't gotten to yet, but I'll explain him in a second, we've suggested that EEG, which would be down here between 10 to the 0th and 10 to the 1 hertz, or under 10 to the 2 hertz, are actually beat frequencies of faster vibrations in microtubules. Now, EEG has been around for 100 years, but we still don't understand its origin and significance of the bands. And we suggested that what it really means is a beat frequency of faster vibrations in microtubules. And I've talked about this with a lot of expert EEG people. None of them buy it, but they can't say why it's wrong. And they don't have an alternative explanation for a systemic approach to EEG. So I just said all this, that a multi-scale hierarchy, and I suggest that consciousness is more like music than computation, that we have these resonances occurring over many, many frequencies in the brain, and resonance interference, beats, and so forth. And if it's a computation, who's looking at the output? That's the problem. So I think it's more like music, and the brain's more like an orchestra. So this is again, the same hierarchical multi-scale system that I suggested. Okay, but why would these quantum processes be conscious? What gives us qualia, feelings, love, joy, emotion, greenness? I was faced with that question after spending 20 years studying microtubules and all the information processing, and somebody once finally said, okay, wise guy, how would that explain feelings, joy, love? The hard problem, as it later became known. And I didn't know, but fortunately that person suggested I read this book by Roger Penrose, The Emperor's New Mind, which I did, and I was quite struck by it and impressed. Basically, he used Gödel's theorem to show that conscious understanding is non-computational, that something outside the computational system is required, and that something was involved with quantum state reduction, collapse of the wave function, and the measurement problem in quantum mechanics. And that, in turn, was a factor related to the underlying structure of the universe, space-time geometry, quantum gravity. Now, this was basically 3 quantum leaps, and the book was met with a lot of derision, a lot of awe, a lot of criticism. But when I read it, I thought to myself, at least this is a mechanism for consciousness. I had never heard of any mechanism for consciousness ever put forth. So I contacted Roger, and we got together. And I'll come to that in a second. But let me first explain one thing, that it deals with the measurement problem, which is that this quantum property of superposition, for example, shown here of a cesium atom, which can be viewed as a definite state in the yellow, or as a wave of multiple possibilities in the quantum in the background. So it can be in multiple locations or states at the same time. until it's measured or observed. And that's the measurement. Why don't we see superpositions in our perceived world? That's the measurement problem. So there are a number of interpretations, and I'll just skip to Rogers, who first dealt with the idea of how things can be in multiple locations at the same time. And he explained that by applying general relativity superposition equivalent to separated alternative curvatures in space-time geometry. So you can see on the left that a two-dimensional space-time sheet. So he came up with these to put it on a slide so you can envision it, and a particle which is basically a curvature. Now, this was first viewed in terms of big masses like the sun with starlight behind the sun that was curved by the mass of the sun. And Eddington did the experiment on a mountaintop in an eclipse and saw the stars behind the sun because the gravitational curvature of the sun had made them visible on Earth. But Roger applied this to small particles. And so a particle in one position would be at the top of A and in the other at the bottom of A, and you could sense them oscillating back and forth. And superposition would then be alternative curvature in basically a separation in space-time geometry. And you can imagine that if these separations were to continue, you'd get multiple worlds of parallel universes, which is a very popular interpretation of the measurement problem. So each possibility goes off and forms its own universe. But Rogers said that these separations are unstable and will self-collapse, undergo objective reduction, that is quantum state reduction by an objective threshold, OR. And the objective threshold was at time t equals h over e sub g, or h bar actually over e sub g, where e sub g is the gravitational self-energy of the space-time and mass separation curvature. And that when this happened, it would be accompanied by a moment of conscious awareness, bing, down at that level. Now, this is the opposite of the observer effect in which consciousness causes collapse. In this case, collapse, which occurs spontaneously by this simple equation, causes consciousness. Now, these would be occurring everywhere, like decoherence in the environment. In fact, you could say that this is decoherence, really, except there are little moments of consciousness occurring, except they're ubiquitous but disconnected and proto-consciousness. There wouldn't be any meaning, there wouldn't be any memory, there wouldn't be any entanglement, and they'd be occurring willy-nilly in the environment, proto-conscious. To get them into our kind of consciousness, we need to do something, and that's where the microtubules come in. And also, these choices may be influenced by non-computable Platonic values. And whatever you think of it, it's the only real mechanism for consciousness ever proposed, as far as I know. Now, Roger needed a quantum computational mechanism in the brain which could biologically orchestrate this information, which is occurring by the Schrodinger equation, would halt or terminate by Penrose objective reduction connecting to space-time geometry with non-computable Platonic values and qualia, awareness and feeling, suggesting these are intrinsic to the universe. Now, this may seem bizarre, but a lot of our leading neuroscientists like Christoph Koch and Giulio Tononi resort to panpsychism because they can't explain consciousness due to computational emergence. This is actually makes a lot more sense than panpsychism because consciousness is a sequence of events rather than a state which doesn't change. So this would be changing from moment to moment, and would also have to regulate neuronal and synaptic activities. So I wrote to Roger and suggested, as Kunal said, that the answer that he was looking for might be microtubules. And we soon teamed up, and after the 1994 Consciousness Conference, we went to the Grand Canyon. There's Dave Chalmers, the philosopher on the far left, Roger in the yellow shirt, me behind him, his wife Vanessa to his left. And we began to cook up our Orch-OR theory. And basically, to jump way ahead, the idea is that there's a quantum bit or a qubit based on quantum dipole pathways in pi resonance groups that would scale up to the whole microtubule and then many, many microtubules. And in a sense, this is something like a topological quantum bit or qubit. And because the microtubule A lattice has Fibonacci geometry, they could intersect and interfere along the way. So we basically proposed that these quantum processes were orchestrated by microtubule resonances and gave it the name orchestrated objective reduction, Orch-OR. So Bing occurs when a certain threshold is met, and what the number of tubulins in superposition will depend on the time t at which it'll occur. So the more tubulins, the larger E sub g, the faster it'll occur. And we used to think that t might be 25 milliseconds, as in 40 hertz, but that would require a very long avoidance of decoherence. We now think that the time is maybe at 10 megahertz, for example, and E sub g would be much larger, but you'd only have to avoid decoherence or premature OR for a 10 millionth of a second. And it's already been shown that microtubules can do much, much better than that. So the basic idea is that the superpositions evolve and resonate in many microtubules, entangle among neurons in different brain areas until threshold is reached at which a conscious moment occurs, because it's all connected at an inward level to space-time geometry, not necessarily out there, but the space-time geometry inside the brain between the ears. And this is occurring at that level. So, yeah?
What is the mechanism that would be causing the entanglement to form?
It could be nuclear spin that I'll come back to later, or it could be the electrons themselves.
So those are specific physical systems that you're saying might be entangled. What would be causing them to become entangled?
Well, some people say everything's entangled. The question is why isn't everything entangled?
But by what physical mechanism, what interaction would cause the entanglement?
Well, you can cause entanglement by a laser causing things to go into coherence, and this whole system is coherence, and microtubules are acting in this way, something like a laser.
Well, coherence is not necessarily the same as entanglement. There's some
Right.
entangled states, or some coherent states are entangled. So are you proposing that there's a laser that creates states in this biological system that...
The microtubules would be the lasers. There's no external laser. They're oscillating coherently. This goes back to Froelich in the 70s. The idea of something like a Bose-Einstein condensate in a biological system due to oscillations in nonpolar regions. Actually, that's how I got the idea to look, but he didn't know about anesthesia. So that's exactly where anesthesia works, due to oscillations in the nonpolar regions inside proteins, and particularly in a lattice protein like a microtubule.
Does your team have an interaction Hamiltonian that would create this entanglement?
We have one that was published in 1993, actually. I'd be happy to show it to you. I don't do that kind of stuff, but somebody did. Okay? So the qualia problem, how do you get qualia? So we claim that the qualia are intrinsic to the fine scale structure of space-time geometry. So the conventional view at the top would be that Bing, the qualia of the redness of the rose, would be to some configuration of activity in the brain, which it is. But the real reason we have the qualia is because we're recreating, or maybe entangling with, the space-time the fine-scale space-time curvature of the redness of the rose out there that recreates in the head. So that's what gives you the redness of the rose, the qualia. So basically, we proposed a sequence of discrete moments, bing, 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 bing. This was originally at 25 milliseconds, but now we think it might be much, much faster at, say, 10 megahertz. But a couple interesting things here, that with each collapse, you can have quantum information going both forwards and backwards in time. And backwards in time would be a very useful thing to have in the brain to avoid the need for post-diction, as Dr. Shimojo would say, or actually rescue conscious free will, because when the way it looks now, we respond non-consciously and have the activity that correlates with the perception to which we respond occurs later. And going back to Libet, if the information is actually referred backwards in time, we can be acting in real time consciously and not be epiphenomenal. There's still problems of determinism and some other things, but at least we can have real-time conscious control. And actually, Roger addressed Ben Libet's backward time effect in his book, The Emperor's New Mind. And I had the pleasure of introducing Roger and Ben Libet at the 1994 conference. So basically, consciousness is a sequence of events, of collapses. It appears continuous, like a movie or a video appears continuous, even though it's a sequence of frames. And the frequency can vary, and if you're excited, the frequency may increase, and your perception of the outer world would slow down. So this time dilation that Shin Shimojo talks about and others talk about could actually be having more conscious moments per time. Michael Jordan said that when he was playing well, the other team was in slow motion, so he might be having more conscious moments than the defense. Well, we got criticized, and everybody said the brain's too warm, wet, and noisy for functional quantum effects. However, and then Tegmark calculated this decoherence time, which he made an error of 7 orders of magnitude, and we found a couple others, and we corrected them in the same journal the next year, calculating 10 to the minus 4th, and 10 to the minus 4th was subsequently shown by Bandyapati, and these events may occur at, for example, 10 megahertz, so you only need 10 millionths of a second, and interfere to give beat frequencies. So how might this work in the brain? Well, conscious perception from vision in this case, for example, goes to the thalamus and then proceeds by 3 waves. You go to the back of the brain to V1 in the first wave, then forward feed forward, and then feed back from prefrontal cortex or somewhere to other parts of the brain. And consciousness only occurs in the 3rd wave. And this was shown, with the 3rd wave. And it was shown by my colleague George Mashur at University of Michigan that only the 3rd wave is inhibited by anesthesia, selectively blocking consciousness. And he showed it for gas anesthesia, for propofol, and ketamine, all 3 types of anesthesia. So anesthesia occurs when you get rid of this 3rd wave. Now, it's the same neurotransmitters, the same physiology. What's different? I'm saying it's because the funny quantum stuff is happening in this 3rd wave and it's not happening elsewhere. And in anesthesia monitoring, for example, spine surgery, we use the 1st wave and the 2nd wave to monitor evoked potentials, but the patient is unconscious. So the brain is active under anesthesia. Then we go to the cortex, and it converges probably on pyramidal cells, which are probably the best bet for consciousness to occur. Could occur anywhere, but pyramidal cells are the best bet. Now, how would this interact with our neurophysiology and choices? And for example, pyramidal neurons in awake animals deviate from Hodgkin-Huxley behavior, and some factor modulates spike threshold on a spike-to-spike basis. This was Nondorf et al. In Nature in 2006. This variability could be modulated from internal microtubules and dendrites and soma. And I reviewed all this, and that shows backward time referral that I mentioned. And it's laid out in this paper I wrote, How Quantum Brain Biology Can Rescue Conscious Free Will. Now I want to begin to close by addressing the entanglement issue and also nuclear spin. This paper at the top will come out in the next issue of Anesthesiology. It's available online already, where they used nuclear spin. They made xenon, an anesthetic, isotopes of xenon, some of which had nuclear spin 1/2. And they found that isotope was 50% less potent than the others. And it had nothing to do with polarizability. They ruled that out. And they concluded that the nuclear spin 1/2 promotes consciousness. The consciousness depends on quantum brain process. Now, I wrote the editorial, which is how I know about this. And this is a battle for 6 months among the reviewers. But it's really an excellent paper. And I tried to put it in the context of quantum consciousness models. They were promoting the idea. They said this does show that quantum mechanics play a role in consciousness, although it's nuclear spin, which is fermions, are different from the boson, the dipole couplings we're talking about. Matthew Fisher has proposed nuclear spin memory by GTP hydrolysis. But how would this affect moment-to-moment consciousness? Nuclear spins can entangle, offering a solution to the binding problem. Now, you probably know the binding problem, and for example, in conscious vision, an object's shape, color, motion, and meaning are processed at different times in different area of visual cortex, yet somehow the disparate content is bound together in unified scenes. For example, a red kite flapping in the wind. Also, auditory, tactile, olfactory, and visual modalities, memory and feelings are all bound together into unified conscious percept, despite occurring in different brain areas and different times. So Einstein's spooky action at a distance, entanglement, made quite literally bind and integrate disparate brain content into unified conscious moments like frames in a film or video. Sequences of such moments can give rise to our familiar stream of consciousness. So I think consciousness is a sequence of events. I'm going to show some conclusions, and then if we have a couple minutes, I just want to talk about taking advantage of this clinically. And the Orch-OR theory suggests consciousness depends on quantum vibrations in microtubules, a deeper layer of the brain's neural networks linked to processes in space-time geometry. Now, I'm not big on AI, but I understand the greatest advances in recent years from deep learning, in which you intersperse convolutional networks. And this could actually be analogous to what's going on inside each neuron in the microtubules, faster, more efficient processing. Anesthesia appears to selectively block consciousness by dampening terahertz oscillations. In the blue light region of the spectrum, Meyer-Overton defines a quantum-friendly environment in living systems. And biological aspects of Orch-OR are far better supported, I claim, by evidence than any other theory of consciousness. Yeah?
What's the relationship between terahertz and blue light?
Blue light is, I said it before, 613 terahertz, 6.1, whatever the next one up. I showed the spectrum before.
Yeah, but
I'm not saying light is emitted. I'm not saying that. I'm saying it's internal. It's not photoexcited. It's within it stays within the system under normal circumstances anyway. Let me just show you a couple of other things that brain stimulation, modulation, including TMS, TDCS, photons, and transcranial ultrasound are used to stimulate the brain. Only TUS can be focused, as shown here. We did the first study of ultrasound on mood in humans. I was the guinea pig, and we published the first paper in 2013. Recently, Jay Sanguinetti has done some more work showing low-intensity subthermal ultrasound improves mood. And I should say ultrasound is mechanical vibrations in megahertz, and we think that they're addressing megahertz vibrations in microtubules. He's also shown enhanced connectivity in the brain with following ultrasound. And this is relevant because Alzheimer's disease, your microtubules fall apart, the tau falls off. And this paper from Australia a few years ago, that in mice at least, ultrasound had a significant beneficial effect on both the pathology and the cognition. And Anirban showed that 2 megahertz stimulation was optimal for prolonging and polymerizing microtubules. And this study by Sadviki and Gupta showed that ultrasound prevented the disassembly of microtubules in an assay. Finally, I want to conclude with memory. Memory, I think, is stored in microtubules. The synaptic proteins that are attributed to memory only last hours to days, and memories last lifetimes. One of the things that happens when the calcium enters is CAMK2 is activated, calcium calmodulin kinase 2, which binds to microtubules. The calcium causes it to transform into this nano-robot-looking thing. And with phosphorylation sites, it can phosphorylate up to 6 sites at a time. And we showed in this paper that this exactly matches spatially and geometrically microtubule lattices, and suggested that CAMK2 encodes memory by landing on a microtubule and phosphorylating up to 6 bits at a time. And this is where memory occurs. So I'll close there and be happy to take some questions.